Hi, everyone. Welcome to D Squared Classroom and Happy New Year. I'm Dori.
And I'm Loren. We wanted to share some of the science behind a very hot topic in the news recently. Genetic engineering is the process of using DNA technology to alter the genetic makeup of an organism. This is a very specific discipline in bioengineering.
And recent advancements in this space have made it possible to successfully implant animal organs into humans. So, today we're going to talk about the basics of how engineers made this happen. Let's jump in.
Xenotransplantation is the process of taking live cells, tissues, or organs from an animal and implanting them into a human. When this occurs, there are steps that need to be taken that allow the implant to function normally in the human body and to prevent the body's immune system from rejecting it. Genetic engineering means genetic modification of a particular cell, organ, or tissue. Scientists have the capability of modifying an organism's DNA so that the animal will have certain traits, promoting success when transplanted into a human body. One technique for genetic modification is called transgenesis, which is the process of adding genes by injecting foreign DNA into a fertilized egg. The egg is then placed into a foster mother where it is allowed to develop to a full term of the pregnancy and born naturally. Transgenesis allows for specific genes to be added. However, sometimes that is not enough. Certain genes might have to be silenced so that a human immune system does not recognize the organ or tissue as foreign. To do this, scientists induce mutations or change the DNA in embryonic stem cells. These modified embryonic stem cells are grown in a science lab and then injected into a very early embryo. This new embryo that contains both normal and modified DNA is again implanted into a foster mother. These genetically modified animals may be allowed to breed, which would pass the modifications to new generations of animals.
Pigs have a very similar circulatory system to humans, so scientists believed that a pig heart would be able to function in the human body. 3 genes in the pig would need to be modified so that the organ was not rejected and attacked by the human immune system. These modifications included knocking out a gene that produced a sugar called galactosyltransferase, adding the human immune system gene that produces the CD46 protein, and adding a human gene that produced thrombomodulin, which prevents blood clots. When these modifications were used to change the pig's heart, the process was tested by implanting the modified heart into baboons. With this test, scientists discovered that the pig's heart kept growing and became too big to function properly in the baboon. The scientists explained the situation as massive cardiac overgrowth. To solve this issue, a 4th gene was modified and scientists turned off the growth hormone receptor gene. This change kept the pig's heart small and optimal for human implantation.
The culmination of decades of work in xenotransplantation occurred just last week when the first human received a genetically modified pig's heart at the University of Maryland Medical Center. The heart was placed in a special machine for preparation before being implanted into a dying man with heart disease. This particular patient had been turned down for a normal heart transplant by several programs and was being kept alive by an oxygenation machine, so the xenotransplant was considered a last resort. The surgery was successful, and as of a week after the procedure, the patient has still not rejected the pig's heart.
This is big news for the world of medical science, but it does raise a lot of questions, both clinically and ethically. We'd love to hear your thoughts on this novel cardiac xenotransplant.